So today we are going to discuss about the third line of defense. Third line of defense is the third line of defense which is used when the first and second line of defense is failed to be uh, allowed to catch the pathogens and kill them. So third line of defense being the last line is very specific, which means the first and second line of defense were non-specific. They were attacking on any pathogen, any sort of foreign particle which attacked our body. But here, third line of defense is very specific, which means it only attacks a particular type of foreign particle, a particular type of antigen, a particular type of pathogen. So, third line of defense here consists of cellular responses and molecular responses. We are going to discuss what are these as well in our next video. Here I'm just going to provide you an introduction of what third line of defense consists of: molecular responses and cellular responses. So we study or we divide the third line of defense in two major parts: the cell mediated immunity, which is CMI, and humoral immunity, so cell mediated immunity involves cells such as T cells, B cells, and macrophages, cytotoxic T cells, and dendritic cells as well. But these are mostly found in the second line of defense. We rarely consider dendritic cells in the third line of defense. Rare. So T and B cells are T and B lymphocytes, and these are developed in the bone marrow. Developed in bone marrow. These are developed in bone marrow, but the T cell is matured in thymus, which is a gland, and B cells are matured in bursa of fabriculus. In bursa of fabriculus of birds is matured in bursa of fabriculus, which is located in the cloaca of birds. Cloaca of birds. So as it is matured in bursa fabriculus, which is located in cloaca of birds, we call it as the B lymphocytes, whereas as the T lymphocytes are matured in the thymus, we name it as T cells or T lymphocytes. So in humans, bursa fabriculus is not present. So instead of bursa fabriculus, in humans, the B cells in humans are matured in pair patches, the spleen, lymph nodes, and sometimes in the bone marrow. So this is the place where these are matured. And when it comes to the macrophages, what are macrophages? Macrophages are matured macrophages are matured monocytes. Matured monocytes which are present where these are present in the non-lymphatic tissues of the body. These are present in the non-lymphatic tissues of the body. Present in non-lymphatic tissues of the body. For example, if we are taking macrophages in immune system, in immune system, macrophages are termed as neuroglia, which further has its important type, which is microglia. When it comes to the use of macrophages in liver, when we use macrophages in liver, then these macrophages are known as Kupffer cells. When we use macrophages in the lungs, Kupffer cells are known as alveolar macrophages. Whereas if these macrophages are present in the lymph node, these are known as the lymphatic macrophages. When it comes to the cytotoxic T cells are types of T lymphocytes which we are going to discuss in our upcoming videos. And the dendritic T cells, as we all know, what are they? These are also matured macrophages. These are matured monocytes. These matured monocytes which are found in the lymphatic tissues. What type of lymphatic tissue? The secondary lymphatic tissues. These are found in the secondary lymphatic tissues. Secondary lymphatic tissues such as the spleen, Peyer's patches, and another name which is tonsil, right? So these are some secondary lymphatic tissues where matured monocytes, dendritic cells are supposed to be found, and afterwards let us discuss about the humoral immunity. In humoral immunity, as we have previously discussed in the concept of monoclonal antibodies, that whenever the helper T cells detect any sort of pathogen, any sort of antigen, then they release interleukin 2. Helper T cells release interleukin 2. They release interleukin 2. As they release interleukin 2, the interleukin 2 allows to activate B cells into the plasma cells and B memory cells. So when the interleukin 2 is activated into activated and transforms the B cells into plasma cells and B memory cells, then this type of conversion, this type of immunity, when the differentiation of B cells takes place, is studied in the humoral immunity, which we'll also discuss in our video, but that is only the intro. Hence, this is what we study in the cell mediated immunity and humoral immunity. Now, let us discuss about what is an antigen. What is antigen? Antigen is any foreign particle which triggers the immune response. Any foreign particle which triggers the immune response is known as antigen. That foreign part that is not supposedly gonna be a foreign particle. That can also be a self antigen. That can also be a self antigen which is present in our body, which triggers immune response, such as in autoimmune disorders. Self antigen is present. Autoimmune disorders such as arthritis, rheumatoid arthritis, self antigen are present, which causes to trigger the immune system. And secondly, foreign particles here do not mean specifically pathogens. Pathogens do trigger the immune response, but alongside pathogen, any other foreign particles such as dust, smoke, or perfume particles or perfume which triggers immune response, which cause allergy or allergic response to the body, causing to trigger the immune system, is known as the antigen. So we also consider dust, smoke, or any particle which triggers the immune system is known as antigen. And how is it considered as an antigen? This is by the help of the MHC. What is MHC? MHC is major histocompatibility complex, which can secrete more than 100 molecules. It can secrete more than 100 molecules such as enzymes, hormones, enzymes, and cytokines. Hormones, enzymes, and cytokines, and many other types of protein which are required to protect the cells from antigen. So these MHC are present and divided into two classes: MHC class 1 and MHC class 2. MHC class 1 acts as a body tag, acts as a tag that this cell is possessed by our own body, so we don't need to kill this cell. It says the immune cells in our body that this cell is our own body cell, so we don't need to trigger kill it or do any sort of action against it. So MHC class 1 is present in all cells of body, in all nucleated cells of the body, except the sperm cells, as well as it is also present exceptionally in the platelets. It is present in platelets, but again that is not present in RBC, which is matured RBC, whereas platelets is even though non-nucleated, but that is having MHC class 1. But you know what, RBC is a mature cell, is a mature non-nucleated cell, but you know what, doesn't contains MHC class 1. Afterwards, when it comes to MHC class 2, MHC class 2 is specifically present in the immune cells, of the body. What type of immune cells of the body? Immune cells such as T cells, B cells, immune cells such as T cells, B cells, macrophages, dendritic cells, T cells, B cells, macrophages, and dendritic cells contain what? They contain the MHC class 2. They contain the MHC class 2, which allows these immune cells, T cells, B cells, macrophages, and dendritic cells to generate a response against the immune response. Now, what happens if the antigen, we have already discussed these things in our previous lectures, that if the antigen is attached to MHC class 1, if antigen is attached to MHC class 1 of what, of the T cells, if antigen is attached to the MHC class 1 of the T cells, then the T cells will themselves produce antibodies, against the antigen, will produce antibodies against the antigen. They will do this thing by themselves. They will not require any other cell to be triggered, or what further differentiation or production of antibodies does not take place. They themselves just produce antibodies against that antigen. But if the antigen is attached, if the antigen is presented to the MHC class 2, the whole mechanism is already discussed in our previous video, so if you haven't watched those videos, you can do check it out on my YouTube channel. So if the antigen is attached to the MHC class 2 of the T cells, then the T cells activate then the T cells activate then the T cells activate other B cells. The T cells activate other T cells. Which type of T cell would be activating? Helper T cell would activate the B cells, and B cells would be again differentiated into plasma cells and B memory cells. Now plasma cells produce antibodies, whereas the B memory cells not produce antibodies, but they store information of that antigen, that this antigen attacked over our body, and if there is any attack in future, then we are going to encounter that antigen with the help of this B memory cell. Now when the antigen attacks again, when the antigen is unfortunately attacking again our body, so the B memory cell will identify the antigen and convert themselves into the plasma cells. If they are encountered. Convert themselves into plasma cells if antigen enters again. If antigen enters if the antigen enters again, of course, if the B memory cells are converted into plasma cells, they will produce antibodies against that antigen, and the person and the antigen would be killed accordingly. So that is all about the introduction of third line of defense. Hopefully we'll meet in the next video, where we are going to discuss about the cell mediated immunity as well as the humoral immunity. Thank you.